So I'm going to talk about the many paths which are possible for artemisinin resistance in Plasmodium falciparum. So to this audience, we don't need to give the introduction to malaria, but we need to keep reminding ourselves that the main obstacle which we are facing in malaria elimination is the resurgence due to drug resistance and the problem of antigenic variation. And because of this, we are not able to make some nice vaccines. So in our lab, the main focus is to understand the genomic and transcriptional basis of drug resistance and antigenic variation in malaria parasites. So we do have a lot of work on epigenetics and transcription, and also we are trying to develop some novel vaccine candidates, which we are not going to talk today. But today I'll be mainly discussing the drug resistance problem. So it's not that we did not have success with a number of cases. In last several decades, the number of cases have come down significantly. But there is some kind of stagnation for last few years. And in 2020, there were 14 million more cases and around 70,000 more deaths than in 2019. Of course, part of it was due to the disruption caused by the pandemic, but part of it was also attributed to the problem of drug resistance. Parasites are becoming resistant to almost all antimalarials, including artemisinin-based combination therapy. Now, for some unknown reason, the resistance has always first emerged in Southeast Asia, then it has spread in India and then to Africa. The first definitive report of artemisinin resistance has come from India in 2018 from Kolkata. So artemisinin is considered as the last line of defense now because we don't have any other drug currently in reserve. It's an excellent drug because it's a prodrug which is activated under oxidative environment. So it's activated more in infected RBCs as opposed to uninfected RBCs. And that also makes it an excellent drug by means specificity. It's very highly specific and killing mostly parasites. There are almost no side effects. But as mentioned earlier, it has a very short half-life. And this is where it is always given in combination with other drugs. If you have to look at its mechanism of action, it is actually it's just alkylating non-specifically maybe a large number of proteins, whatever is in proximity. And this is where it's not really very specific as a target, means there are no specific targets. Whatever you will find, it will be alkylated and will be damaged by artemisinin. Now, when the first report of resistance came from Southeast Asia, a lot of genotype-phenotype association studies were performed, and a large number of sequencing were done for the samples which were resistant, and it was found that CELCH13 essentially emerged as a molecular marker for the artemisinin resistance. Using reverse genetic engineering, again, it was demonstrated clearly that this is a gene which is a definitive marker for artemisinin resistance. So how does it work? CELCH13 is an adapter to the ubiquitin ligase, and what is believed is that because artemisinin is causing a lot of damage in the system, many proteins are damaged and they are ubiquitinated and are subjected to proteasomal degradation. It kind of there's a lot of system basically has a lot of protein damage and it is not able to recover with that, and that's where somewhere cells die. So to slow down this process and to enter into some kind of dormancy, the ubiquitination, if there is a mutation in CELCH13, will lower the ubiquitination of the damaged protein. Cells will buy time, they will upregulate some stress response pathways, and this is how the mechanism for the CELCH13 mediated resistance was proposed. Later, another paper came which indicated that CELCH13 is also present at the mouth of the cytostome, which are basically involved in uptake of hemoglobin. Now, because it is associated with the hemoglobin uptake, if there is a mutation in CELCH13, there will be lower hemoglobin uptake, lower activation of artemisinin, and again, it will confer resistance to the artemisinin. So these are the two mechanisms proposed by which CELCH13 is conferring resistance. So to begin with, we started to ask the question, what are the other co-selected mutations which can be complementary or compensatory in nature present along with the CELCH13 resistance marker? So we analyzed more than 2,500 genomes, and we asked the question, what are the mutations which are always coexisting with the CELCH13 mutation? So here, if you look at this graph, what you'll clearly see here, there are a few things which can be, we can point out clearly. One is that C5ty, which is a CELCH mutation again, is a major marker of artemisinin resistance. you will see that majority of the parasites which are having Kelch mutation have this particular mutation in it, indicating that system has reached or reaching almost some kind of neutral fitness with this particular mutation. Another thing you will see is that wherever there is a C580Y mutation, you will see in this area there are no other mutations in the Kelch protein, indicating again that parasites cannot tolerate more than one mutation in Kelch 13. You can see this clearly here also. There are other Kelch mutations, but they are kind of mutually exclusive. So there is a fitness cost associated with Kelch 13, and parasites can survive up to only one mutation per one protein. Right. Now, when we looked at other mutations which are coexisting, so when we say coexisting, they are there in almost 80% of the resistant parasites, but at the same time, they are absent in 80% of the sensitive parasites. Two genes which were coming up in this case very strongly are, first is an alpha-beta hydrolase, a lipase enzyme. Second is an HECT or HEC domain containing protein, again, an adapter to the ubiquitin ligase. These two are basically are present in almost all resistant parasites and absent in almost all sensitive parasites. Another gene which has been co-selected is ATG18. ATG18 is an autophagy protein, and before Kelch 13 was considered as a molecular marker, people have also proposed this as a marker for Artemisinin resistance. And the reason for this was that it was always present in resistant parasites. And none of them were incorrect, because when we did this analysis, wherever there was Kelch 13 mutation, ATG18 was also there mutated in those parasites. What was interesting is that when this mutation, T38I, was introduced in wild-type parasites, instead of growing or having any effect on the growth, those parasites were growing faster. So what it is indicating is that Kelch 13 mutation is causing a fitness cost, and that is compensated by other complementary mutations like ATG18, which are then facilitating the higher growth. So in the process, we have also basically, it suggests that ATG18 is a complementary or a compensatory mutation in nature, but it is not by itself causing resistance to the parasites. In the process, we have also demonstrated that autophagy underlies the proteostasis mechanism for the Artemisinin resistance. Now, what about the parasites from India? So, as I said, the first report of Artemisinin resistance has come from India, from Kolkata. So, we approached that group, and with them, we performed large-scale whole genome sequencing. 71 we are calling large-scale because before this, there were only 5 Plasmodium falciparum genomes sequenced from India. So when we did the sequencing, to our surprise, there were no Kelch 13 mutation in any of these parasites. Now remember, this is not a single report now coming out from Kolkata. There are several reports coming out from India and Africa which are indicating resistance, but looks like it is a Kelch 13 independent resistance. It's not dependent on Kelch 13. If you look at the phylogeny of the parasites which are coming from India, so this is what the Indian parasites are. They are somewhere closer to the countries or the parasites which are coming from the African isolates. And they are a little bit away from the Southeast Asian population, indicating that we have a different genetic background, and the resistance which is developed on that background is different than what we are seeing from Southeast Asia. And this is where Kelch 13 is not an ideal marker for Artemisinin resistance for parasites from India as well as from Africa. What was again interesting is that the genes which we found in our earlier screen, which were coexisting with Kelch 13 mutation, some of those genes were identified in some percentage of the parasites in Indian isolate. So for example, another HEC domain containing protein was the major gene which was there in around 10 to 15% of the parasites. Alpha-beta hydrolase, another lipase, which we have demonstrated now, is also mutated in a large number of parasites here. So what we believe is that ubiquitination, so in this case, Kelch 13 is also causing, means it's involved in ubiquitination. So it's the process which is more important for the resistance rather than the gene itself, because we are keep seeing ubiquitination and the vesicular transport pathways as the major targets for the Artemisinin resistance. We are following up with this work now. We have started working on these proteins to see if they are really indeed markers. So we are now making mutations in the wild-type parasites to see if they can confer resistance to the wild-type parasites. Now, in parallel, there are several reports which are indicating that Kelch 13, yes, it's a definitive marker for Artemisinin resistance, but you can have parallel mechanism of Artemisinin resistance. You can have stress responses playing an important role in Artemisinin resistance. So for example, particularly the oxidative stress responses and protein damage response pathways are shown to be independently driving the artemisinin resistance. There are also some kind of propositions that there might be involvement of some transcription regulator, which is actually involved in the regulation of this stress response pathways. As our lab is mainly focused on epigenetics and transcription regulation, we started to look for the some kind of a global regulator of stress responses in Plasmodium falciparum. And as we had some experience working with hierarchic systems, we know from there that GCN5 is a global regulator of stress responses from yeast to hierarchic systems. We asked the question, what is the status of GCN5 in Plasmodium? It is conserved. It has historical transferase domain. It has bromodomain, so all the domains are kind of conserved. It has some N-terminal, much larger region, which we don't know what it is doing. So we asked the question if GCN5 playing any role in stress responses. So we took parasites, we subjected them to various kinds of stresses. We are not going to the details of stress. Then we performed RNA sequencing and chip sequencing for GCN5. First thing to note here is that GCN5 is always upregulated in under almost all stress conditions. So it looks like has some role there. Under these stress responses, one thing which was very evident is that there are some proteins, some of the saprons, which are commonly upregulated or deregulated in almost all stress conditions. Genes which are upregulated are mostly important for cellular homeostasis during stress conditions. Now, genes which are upregulated are those also associated with GCN5. So we have done chip sequencing, and we did see that genes which are associated or upregulated under stress conditions are all bound by GCN5. So you can see here that these are overrepresented as opposed to the control conditions. This is to show here all the pathways which are shown by the transcriptome analysis that they are upregulated in artemisinin-resistant parasites. And again, if you compare them with the sites which are bound by GCN5, there is a huge overrepresentation binding of GCN5 to genes which are associated with artemisinin associated stress responses. So now we wanted to see what if we inhibit GCN5. Unfortunately, we could not make a knockout or knockdown. We did use but some inhibitor, and on the recombinant protein, we showed that this inhibitor actually inhibits GCN5 very strongly. So when we treat the parasites with this particular inhibitor, and if you look at some of the BIP and trich saprons, which are known to be actually as some kind of markers of stress responses, you do see that they are upregulated under stress conditions. But if you treat the parasites with now inhibitor of GCN5, you see that their levels are going down. Now, what about the inhibition of GCN5 in parasites? So if you take a concentration of GCN5 of garcinol, which is kind of sublethal, which is like having no effect on the parasites otherwise, but now if you take this in conjunction with artemisinin exposure, you do see a significant decrease in the survival of the parasites, indicating that they are now becoming more sensitive to artemisinin if you have inhibition of GCN5. If you take higher concentration of garcinol, if you inhibit much higher at higher level GCN5, you do see a complete reversal of the sensitivity of these parasites to artemisinin. So what we found is that PF-GCN5 is upregulated under various stress conditions, also in artemisinin-resistant parasites, and it regulates stress and stimuli-related genes. And its inhibition can basically reverse the sensitivity of the parasites. This was the first report of chromatin regulator playing any role in artemisinin resistance, and of course we had a lot of resistance there, and we had really hard time publishing this work. But what is interesting is that other groups are now coming up with almost same observation, where they do see that Plasmodium falciparum GCN5 plays a key role in regulating artemisinin resistance related stress responses. So what we know now is that in the sensitive parasites, PFKH13 is involved in endocytosis of hemoglobin. And this is where artemisinin also get accumulated and get activated. PFKH13 is also involved in the ubiquitination of the misfolded protein. And in the resistant parasites, the endocytosis goes down, which leads to lower activation of artemisinin. And at the same time, the unfolded protein response, which like the proteins which are misfolded, will have lower level of ubiquitination. At the same time, it will increase the PIP3 vesicularization. Autophagy goes up, and what we have also shown is that the stress response pathways kicked in, means that was already known, but we showed that PF-GCN5 is involved in the regulation of those genes, And in another work, we have also shown that PFH type 1 is actually working on the other side of this regulation. So this is what, for the time being, we know about the artemisinin resistance. But the question is, does it explain all kind of resistance we see in the field? And the answer may be not. And I'll try to explain that here. So let's say these are some of the cell lines which are known for the artemisinin resistance. And artemisinin resistance actually is calculated by ring stage survival assays, where some percentage is given based on the parasites which are able to survive. So if you compare now these two lines, which are having exact same mutation, which is C5TY. So they have only a single mutation in Kelch protein at C5TY position. And in one line, there is 10% resistance. In another line, there is 30% resistance. Now, if Kelch13 is a definitive marker, how do you explain these differences in the resistance level? For example, we all do transformation in E. coli. You put a plasmid. If there is a gene for resistance, all bacteria should survive wherever there is a gene for resistance, indicating that there is a complex genetic background. It's not a universal identifier or determinant, Kelch13 by itself, for the resistance level. There are other genetic mutations which are playing some role in determination of the resistance level. And this is where the complementary or compensatory mutations which we talk about play a role. But that would still explain the other genetic mutations. Now you take any one of these cell lines. So let's take the last one, which is C5TY, and it has around 30% resistance. Now in this case, it's an isogenic background. All the parasites have same genome. They're all growing under same environmental conditions. But if you now expose them to artemisinin, only 30% of them surviving. So what happened to the rest of the 70%? If genetic background is the key factor in artemisinin resistance, why not all of them are surviving? And this is where we think that transcriptional heterogeneity or the population level heterogeneity is playing a significant role in artemisinin resistance. Now, in a multicellular system, heterogeneity is easy to explain. You have different cell types, and this is causing cells which are changed due to some conditions. But in a unicellular system, it's little complicated. It can be also a mean for survival, right? So, for example, if there is a more diverse population, the chances of survival would be higher in a unicellular system. There are two possibilities. One, that all the populations which are eventually emerging resistant, let's say in this case, are all present to begin with. And that is called something called bet hedging. So basically all the subpopulations are present. You subject the population to a particular drug, and some populations survive because they have those kind of favorable genetic background and something beyond. Or there is a possibility of acquired drug resistance. So they do acquire those possibilities when you subject them to that particular environment. So we ask the question, what is it in Plasmodium? What is the heterogeneity in the Plasmodium? So we perform single cell RNA sequencing to identify the population type in an otherwise morphologically single synchronized culture and see what is the transcription status of those. So we used 10x genomics workflow. I'll not go into the details of that. What we have done is that we took highly synchronized parasites. We have done double synchronization because our goal is not to see different stages of the parasite. We are not looking into the morphological stages. What we are looking for is morphologically similar parasites, but transcriptionally different, if there is any. So we are interested in transcriptional heterogeneity. And further, to understand if any stress can cause change in the heterogeneity, we subjected them to temperature stress, which basically is mimicking the cyclic bursts of fever, which we see during malaria. We have sequenced around 10,000 cells, and first thing which we noticed is that, yes, there is a lot of heterogeneity. So you take, let's say, these parasites and you put them under the microscope, almost all of them will look same. But if you look at the transcription of those now, there is a lot of heterogeneity in them. Second thing is, if you subject them to temperature stress, there is a population emerging, which is there only under stress conditions, but not there in normal conditions. Also, there is a lot of rewiring of the transcriptional program. So the populations you see here are also changing when you put them under different stress conditions. Another thing which is there in the field for a very long time is that when there are no nutrients and all kind of unfavorable conditions are there, parasites are converted into gametocytes. But looks like that's not true. Parasites are converting into gametocytes almost at every cycle, because here you see that there is some kind of cell-fate decision when they are converting into gametocytes when the conditions are all favorable. And still 5% of the parasites are going to the gametocytogenesis. What happened under temperature stress conditions? I think there's some problem with this, but the markers of gametocytogenesis are further going up when you put them into under temperature stress conditions. So it's facilitating, but they're already going for that particular path. And it does make sense from the parasite perspective because it does not have to wait till end to convert into the gametocytes. It's keep converting into the gametocytes. So whenever there is a bite, there is some transmission, right? Now, anyway, our goal is to look from the heterogeneity perspective. So we also perform bulk RNA-seq of these samples. What we can clearly see is that temperature treatment leads to decreased global transcription, but it increases the heterogeneity for the genes involved in chaperone activity, gametocytogenesis, and maintenance of species. All of this will suddenly, basically, these are the all words which were coming from the artemisinin resistance also. These are all playing important role in artemisinin resistance. So we ask the question, what is the status in all these cells of stress responsive pathways? So to our surprise, what we see is that, again, remember, this is all grown, as he said, under very favorable conditions. Nutrients are given in plenty. There is no limitation of those. Temperature is fine and under all favorable conditions. Again, even in those conditions, around 10%. So this is like only this cluster, 5, 6, and 7, which are very small proportion of the total population, are still expressing very high level of stress responsive genes, indicating that there is some kind of a persistent population which is already always present in the all population, and it's possible that this is the population which is first responding to any unfavorable condition. So in this case, let's say all these cells have Catch13 mutation, but maybe this is the population which is first responding to the artemisinin exposure and eventually emerging as the resistant parasites. Just a few more minutes, 2, 3. Okay, so this is one way to generate heterogeneity. Is there any other way? In cancerous cells, RNA editing is another major way of generating heterogeneity. Or it can be also by transcriptional errors. And what we found in Plasmodium is that it has an order of magnitude higher transcriptional error rate than even in bacteria. So for example, bacteria are universally considered to have very high error rates. But what we found is that Plasmodium as a species has much higher error rates, and that is true for Vivex and other species. What is the physiological significance of this? To cut the long story short, we think that this is again introducing some kind of heterogeneity to the system, right? So you have gene expression level heterogeneity and you have transcriptional level heterogeneity. This kind of heterogeneity, which is coming from the transcription, can provide you the plasticity, which is for the time scale shorter than the replication times, and basically can be another mode of bet hedging. So in conclusion, what we think is that there are many paths to artemisinin resistance. Catch13 is a definitive marker for artemisinin resistance. Other complementary mutations or compensatory mutations like ATG18s play an important role in resistance generation. But more than that, we think that it is not about Catch13. It is about the ubiquitination and the vesicular transport pathways, which are playing an important role in artemisinin resistance generation. Other transcriptional regulators like GCN5 work in conjunction with Catch13 and help in resistance generation. Transcriptional heterogeneity at both gene expression level and at the level of the transcriptional errors also help in generating resistant parasites. And this is the review we have now written, which was invited and submitted in Trends in Parasitology. This is the group we have, and we work at ICGEB Pune. I'm also thankful to all our collaborators. We also had a collaboration in France, and I'm also thankful to the funding agencies and the ICGEB Pune for all the facilities.
Quick short questions.
So, besides Calch protein, in fact, we have finished with the full vacuole proteome, and we found many novel transporters, and we have few more transporters which we believe that they are linked to artemisinin resistance.
I want the students to ask.
Did you find anything?
We have not looked specifically for transporters, but we have a list of like 370 mutations, and definitely we can go back and see if there are any transporters.
We can discuss about that.
Sure.
That and the third is there are ubiquitination pathway. You know what are the proteins which are actually associated with artemisinin resistance? Because we published one paper where we showed that shuttle proteins are actually associated. One other DDI, that is also DNA damage control protein. And we think it's a very good target again.
As far as I believe that for artemisinin, there is no selectivity or specificity in targeting. Whatever it can find in proximity, it is just alkylating and leading to protein damage. And this is where you will see targets everywhere, like in proteins, in DNA, everywhere. It's, I think the mechanism is coming from the how they are responding to those damage pathway. So, yeah. Mr. Friends, any question?
Any question from students?
Yeah.
Yes, somebody.
Yeah. Yeah, please.
One question.
Yeah, please.
So you said with your GCN5, you
Yeah.
reversed it.
Means we inhibited it and they are now sensitive to artemisinin.
So you try to do single cell RNA-seq on them.
No, not.
What is changing and how many are changing?
No, we haven't done that. Single cell, unfortunately, is very expensive. So
I know, but
cannot be done with like RT-PCR or PCR, but yes.
and also another one when you said that
No, both
population
Come data.
Sorry, I can't hear.
growth has changed. The growth rate has gone up.
For the ATG18 T38I mutation, so it seems this particular threonine is phosphorylated and it leads to reduction in autophagy and growth. So when it is mutated, it is leading to higher growth. So this is what was shown earlier. But what we are now seeing is that we can put these two pieces together. So ATG18 is actually compensating for the fitness cost, which is associated with the less hemoglobin uptake.
But you know where the growth is affected, which type of that growth.
No, we are talking overall growth. So we are just talking parasite growth. So I'm not really sure which.
Trophozoite. Or,
Okay, maybe I can go back and.
I mean, cell cycle part, there are different parts. So which part?
We are, I think it's overall cycle parasitemia as such. I'm not sure which.
Do you think that the cell cycle is shorter or longer?
It is just about the protein homeostasis. Now, let's say nutrients and proteins were not that much available when there is a KET13 mutation. But if there is also ATG18 mutation, it is compensating from there. So that's what we.
Okay, question from those to that question. If the growth rate is faster, that means total cycle 48 cycles.
No, means parasitemia was going up.
Parasitemia increased per
Parasitemia
cycle.
increased. So, for example, in one cycle you are getting 10%, it is now 12%. So, in that sense, otherwise.
More merozoites are being formed, you mean?
So, this is not our work. I'm just so this is an older work from some other group when they first proposed ATG18 as a marker of artemisinin resistance.
Okay.
But when they make the genetic mutation, they said it is going faster. So, I don't think they've characterized at what stage it was faster.
Okay,
Since I've heard ATG18 many times and be sure it.
so maybe more discussions over lunch.
You can start your talk if you can get it started. So ATG18 is known to bind PI3P, and Kasturi and Souvik have shown that PI3 kinase contributes to KET13 target and so on and so forth. So there is almost a conspiracy theory going on there. Although what is a little bit more challenging to prove, I can put it more politely, that is if it is a PI3P component, which is actually adding to the resistance bit and so on and so forth. But since ATG18 interacts with PI3P and then
Unfolded protein responses and means some of these actually they work in conjunction. So this, if it is upregulated, that also will upregulate the other pathway, which one was that? I think autophagy and PIP3 vasculation. So both are actually working in some conjunction.
Okay, thank you very much.
Long live India and France friendship.